So chapter 3 is cell structure and function. We are not going to go back through all of the structures and functions of cells that you learned in your 1406 biology. And so we're going to focus on things that are specific to the microorganisms, and especially things that are important for identification of different organisms from each other, for distinguishing organisms under the microscope and with different biochemical tests and so on. So that, because identification is an important part of the course. So you're expected to know that prokaryotic cells have a cell membrane, a cell wall, cytoplasm, ribosomes, and then DNA that's not enclosed in a membrane, so no nucleus. Eukaryotic cells contain a cell membrane. Some types of eukaryotic cells have a cell wall, some don't. Then cytoplasm, they do have a nucleus, ribosomes, endoplasmic reticulum, smooth and rough, Golgi, peroxisomes, lysosomes, and so on. So those basic things we're not going to go over again. And if you've forgotten some of those things, you can look in your textbook and they do review all those again. But we're going to spend our lecture time, with some of the things that are very specific to certain types, primarily bacteria we'll be focusing on quite a bit. And we're not going to talk much about the archaea, mainly because none of the archaeans have ever been identified that are pathogens that cause disease. So we're going to focus on the domain bacteria, and that's primarily our focus for this chapter. All right. So this list of study objectives, you want to use this when you finish the lecture, when you finish reading the textbook, and so on, to practice for the test. These are the examples of cells that your book provides images for. For example, on the left side, A, you have, these are bacterial cells. These are called rod-shaped cells, and we'll have a more correct name for that in a little bit. And then, but these are bacteria. So you see quite a few of them. This image is taken with a scanning electron microscope. So this is called a scanning electron micrograph. And we will be talking about types of microscopy. But this microscope can measure or can view items that are very, very small. 1 micrometer, you can see, is about the length of each of these cells. So these are bacteria. On the right side, for B, you see a paramecium. This is a eukaryote. And this picture was taken with a light microscope. LM is light micrograph. You can see the scale here is 40 micrometers. So this cell is probably 120, 150 micrometers across, much different in size in real life compared to the picture on the left. So this is a paramecium. A light microscope will have color sometimes in the image. However, some of that is just based on the way the light comes through the specimen and through, depending on if you're using different filters. So sometimes the color that's in a light micrograph is not true to life, but sometimes it is. For any kind of electron microscope, the image always comes out black and white, and then any color that's there has been added artificially. So these bacterial cells, these rod-shaped bacteria, are not truly orange, but the color is added to make it easier for you to see. So like I said, prokaryotes, it's understood that you already know they lack a nucleus. They do have DNA. The DNA is found in a region that we typically call the nucleoid. So that's where the DNA is sort of bundled up, but there's no membrane surrounding it. There aren't any, other than the plasma membrane or the cell membrane, you don't have any additional internal membranes for the most part. There are a few little exceptions to that rule, but for the most part, you don't. So most bacteria don't have it. Most bacteria are somewhere in the 1 micron area of length and diameter, maybe 1 to 5 microns at the most. And so, like I said, we're going to focus on the bacteria, but the archaeans also have these characteristics. So these are the general characteristics of prokaryotes. I assume you know what a ribosome is, what cytoplasm is. We will talk about cell wall, but I do assume you know a little bit about that and cytoplasmic membrane. But we will focus on things like inclusions, which you probably haven't talked about before. We'll talk about different aspects of flagella, and we'll talk about, for example, this thing called Glycocalyx. So we're going to be adding, like I said, to the things that you know from your previous coursework. And just quickly, here's an image of a eukaryotic cell. You would be expected to know about the nucleolus, the ribosomes. Nuclear envelope is just another name for the membrane surrounding the nucleus. Pores, lysosome, secretion, Golgi. You probably know about centrioles. So most of this you probably know about. We will talk about some of the external structures, cilia, and flagella, and for cells that have a cell wall, we'll talk a little bit about that. So one thing about eukaryotic cells is they're noticeably larger than prokaryotic cells, and they can even be more than 100 micrometers. And you'll remember the list that we made in chapter 1 of some of the eukaryotes that would be important for us. Especially the ones that cause disease. The pathogens are going to be the protozoans, the fungus, and some small animals. Algae, there are some pathogenic algae, but we won't discuss them. And I don't know of any pathogenic, at least not to humans, pathogenic plants. I guess there's plant products, but plants themselves don't cause disease. All right, so let's get back to bacterial cells. We're talking about domain bacteria. And the first thing we want to talk about, just external to the cell wall, is for some species, there is a structure or a substance called the glycocalyx. I think we'll call it a structure. It's made up of polysaccharide, which you'll recall is carbohydrate. It can also contain polypeptide, which is in the protein category, and it might have both. So the polysaccharides are what make it very sticky. Actually, polypeptides can also make it very sticky. So it's a very, some kind of, we say gelatinous, kind of like gelatin, sticky substance that is outside the cell wall, but it's creating a layer on top of the cell wall. And there are two types of glycocalyces, which is the plural of glycocalyx. The capsule, which is a little more kind of tight, and not tight, a little more structured and close to the surface of the cell. So we say more firmly attached to the surface, a little bit more organized. And the function of the capsule is to prevent bacteria from being recognized by a host immune system and possibly prevent it from being destroyed and cleared out by the host immune system. So we think the capsule, we know in some instances for sure, the capsule allows the bacterial cell to evade the host immune protection. So evade is similar to avoid, evade. Now, there's another type of glycocalyx, and typically a species will either have a capsule or a slime layer or neither. So it's possible to have neither, but some species have what's called a slime layer. And just like it sounds, it's more of a loose layer, much more water soluble. It extends out further and it's not a tight structure. It's a lot more sticky, and the function of the slime layer, if a species has the slime layer, is for attachment. So we are going to talk about, there are structures that bacteria have that increase their ability for some species to be able to attach to some kind of surface. And that can be very important in how they cause disease. So right now we're just learning about the structure itself. So these would be electron micrographs. So images taken from electron microscopes. And the T stands for transmission, so it's going to give you a very two-dimensional image. And then these are your scales. So this is less than 1 micrometer scale. So 250 nanometers is 1/4 of a micrometer. So yeah, you can see it's about 1 micrometer across probably in the end. But this, what we're focusing on here, and remember, this is an artificial colorization on part A. But this, what they're showing here is sort of this, I guess, fuzzy looking orange layer here between the lines I'm drawing this. That's the capsule for that cell. So that's kind of what it would look like under the microscope. And then they colorized it so that you can see it a little more clearly. For this cell on the right, here's the cell. You can see the cell wall very here, cell membrane. And then outside of that, this whole bit, this whole bit is the slime layer, and it's sort of fallen off a little bit. You can see little chunks of it. So it's a much looser, stickier layer of material. But in general, we're just going to just see roughly the difference between them. And roughly speaking, they're made of the same kind of material. It just depends on how structured that material is, the polypeptide and the polysaccharide, which structure is made, and then what the function is. So capsule is for evading immune response. It's a layer of protection to protect from the immune cells, for example, if it's a human, the white blood cells. And for the slime layer, it's more about attachment to a surface. All right, you also have flagella. Flagella, you're probably familiar with. Bacteria can have flagella, and they're made up of microtubules. We're not going to get into the details of the structure. They're hooked into the cell wall and the cell membrane, but then they extend out of the cell beyond the surface of the cell. Not all bacteria have flagella, but when a bacterium that does have flagella is said to be modal, that means it can move. So it has the ability for locomotion. That's another way to put it. Okay. What we're going to focus on with flagella primarily are what we call the arrangements, because this can be seen under a microscope and therefore used to help identify a microorganism. And so I really want you to focus on these arrangements here, because this will be the words that I use. Your text adds some synonyms, but let's just focus on these. Monotrichus is when a bacterium just has one flagellum at one end of the cell. That's probably the way you're used to thinking of it. Amphitrichus, some species have two at either end. That's Amphitrichus. Lophotrichus is like a bunch at one end, kind of a little tuft of many flagella. And it can be 3, it can be 5, it can be 10. There's not a particular number it has to be, but they're all sort of bunched at one end. And Peritrichus is when there's flagella all around. All right, so those are the words that go with the flagellar arrangements. And of course, you can have a cell that has no flagella at all. Okay, so I'm going to break the lecture here, and you can move on when you're ready to the next lecture segment.